images with deep learning. And before I begin, I'd like to really emphasize, so over the past couple years, I have been incredibly fortunate to have received great mentorship from scientists at Hopkins, at the NIH, and from many other people. And as part of that, I was able to get involved in these exciting projects. So the first point I'd like to make, hopefully one that I don't have to work that hard to convince you of, is that medical imaging and deep learning are very synergistic. So over the past 2 decades, the interest or the amount of publications in medical imaging have been increasing at a steady rate, but really over the past 5 or 10 years or so, do we see an exponential growth in deep learning. And so experts refer to deep learning as a paradigm shift, the data-driven paradigm shift, which implies that it has far-reaching applications in every field. But I think the impact of deep learning in medical imaging is particularly important because it goes both ways. And so what I mean is, of course, you have deep learning models that have clear applications in medical imaging for disease classification and so on. But you also have, in the other direction, problems in medical imaging like interpretability or lack of data in some training cases that then motivate new algorithms in deep learning. So that's what I mean, the interactions go in 2 directions. Just to share with you what I feel are some exemplary AI applications that have been developed by the lab I am part of at Johns Hopkins. So on the far left here, we have what's called image super resolution. So in this image at the top, this is visualizing neurons in a mouse brain as it is navigating through a maze. And so really what's being visualized here is the calcium concentration in the neuron, which is directly correlated to neural activity. And so imaging, there's a common theme, right? There's always a trade-off between 2 factors. If you want fast imaging, then sometimes you have to sacrifice resolution. But that might not have to be the case if you're able to, in real time, apply a deep learning algorithm that can upscale the image. And so the top is the original image, and the bottom is the upscaled image done by a neural network. The next application in the middle, and one that I've worked on and I will present very shortly, is real-time cancer segmentation. So during surgery, you have the surgeon with an optical probe in their hand, and they are examining the region of the brain which may contain cancers. And so this outputs in real time a color-coded map which shows you which regions are cancerous, which regions are not cancerous. And so these types of applications have direct translational potential. And the third one on the right, we have pretty interesting application and different from the other 2 called the self-supervised image compression. So on the left-hand column, you have the original image, and the right-hand column, you have the image compressed by, I believe, 80 times. And basically, probably very hard to see, but the white arrows here are pointing to regions in the image to show that even after compression, important structure in the image is still maintained. So this is usually, so the word self-supervised means that sort of you would use a sort of autoencoder architecture to perform this task. Whereas for cancer detection, the inputs are clearly labeled. That meaning it's labeled cancerous or non-cancerous. For this type of image compression, the input and the output are the same exact thing. You want the network to sort of learn a representation. Now, before I get into my specific projects, I would like us all to enter with an understanding sort of why AI is so powerful. So let us consider that without loss of generality, we have 2 classes, the green square and the red circle. Now, and then on these 2 classes, we're able to measure the value F1 standing for feature 1. Now, if you already observe good separation between your classes, then you can apply the most simple type of classifier, which is just thresholding. You can say if your value of F1 is above or below that threshold, then you can assign a class. Now, it would be nice if all classification problems were so simple, but that's not the case. So maybe we need to extract a second feature, let's call it F2. And really, in a sense, by adding that extra dimension to the visualization, it sort of spreads out that data. So you might be able to have clearer separation. Then you can fit a line between that as the decision boundary. And you can see where this analogy is going. In 3 dimensions, the classes become clusters in 3-dimensional space, and the decision boundary becomes a plane. And this goes on for, although it's not easy to visualize anymore, 4, 5, 6, indefinite number of dimensions. Now, the problem I want to pose is, what if there is a problem that is sufficiently complex that extracting 3, 4, 5, 6, even 7 features is not sufficient? And certainly those problems do exist. In a sense, the problems that are able to be solved by thresholding, where just a limited number of features are considered low-hanging fruit. And so to speak, the low-hanging fruit has already been picked. So we are left with some difficult problems that we need very high-dimensional features for. And so I think this is where one of the strengths of deep learning come into play for 2 reasons. One, deep learning can implicitly learn a feature representation. And number 2, that this feature representation can be very high-dimensional. And as an aside here, some of you may be familiar with ImageNet. So ImageNet models can be VGG, ResNet, EfficientNet, but regardless of all of the model architecture, what they have in common is that after the convolutional feature extraction, they end in a fully connected network. And right before the probability output prediction of these ImageNet networks, you have a vector that is 2,048 elements long. And so what you do, if you pass in thousands of images into a network, get their feature embedding, and then apply some dimensionality reduction technique, you will clearly see different clusters in the sort of like TSNE plot, if you're familiar with that, that show the clusters of the different object classes. So I think that is one famous, very effective example of how a neural network is able to learn high-dimensional and high-level features. Great. So the first application is detecting cancer from optical imaging of human brain tissue. And so the physical model behind this application is that high-grade brain cancers exhibit lowered optical attenuation. And the relationship, I think, is fairly straightforward. What happens when cancer proliferates in white matter, you have degradation of myelin, which surrounds the axons. And so after that degradation, the ability for that tissue to block or attenuate the incoming light is reduced. And so the way we measure attenuation, so the equation up here, you have intensity as a function of depth, and that is an exponential decay with coefficient mu, called the attenuation coefficient. And if we can linearize that by taking the log of both sides, so this value we measure, the value A of z, we can measure ex vivo, and then we have the depth. So we have all the information we need, we can just fit the slope as the attenuation. And so here is, using this method, so the histology slide it has labeled where the high-grade cancer is and where the low-grade cancer is. And on the left-hand side, there is the corresponding attenuation map, where the red is corresponding to low attenuation. So you can see clearly there is a good correspondence between histology and this attenuation mapping method. Of course, this method is highly interpretable. We have a robust model behind why this is supposed to work. But what it doesn't do is it doesn't take account local information into the prediction, meaning that for each value in the scan, we calculate an attenuation and we just threshold that, regardless of the attenuation values of the surrounding region. And so that's why we turn to deep learning, which can utilize spatial features at the cost of interpretability. So as a researcher, my job here is to sort of explore different solutions to important problems. Reasonably, one might say that you would rather have the interpretability, and so I think that's perfectly reasonable, but here we want to present as many different solutions as possible. So what I do, I have 12 patients. I split them into a training and testing set, and then I leave one just to do some visualization, segmentation at the end. From each patient, we extract several tissue samples from their brain that are then evaluated by a pathologist. And so from there, so for each tissue sample, after we image it with the optical coherence probe, the scanned volume looks something like this, right? Where the top, looking down from the top, it's about a 2 by 2 millimeter region. And so our goal is to turn this into a coded map where it tells you, in this region, is it cancer or non-cancer? And the way we do that is by splitting the entire volume into little slices, which are outlined in red here. So for each frame, we split it into several slices. And so the goal is that once we have the trained network, we can pass this in slice by slice and then tile all the outputs into a segmentation map. So here are just some visualizations of what the data looks like. So these are the slices, and then from there we also generate a corresponding texture. The texture we use called the gray level co-occurrence matrix. Some of you in computer vision may be familiar with it, but it's a very common way to extract texture. So basically, we take this sample of, we have this sample that we scan of the patient tissue, and then we, from that, have the slice data, and we have the texture data, and now we train the neural network. And just, by the way, so Wang, that's me. So this project is currently in review for Biomedical Optics Express, so currently in peer review. So here's the model architecture that we use in this project. So I'll break this down. So first thing to notice, so we have actually, this is not one convolutional network, this is actually two of them joined together. So hence why we say ensemble network. Let's focus first on the blue one at the top. We train to classify cancer based on the slice, which I just presented to you in the past slide. And then we train a separate network to classify the presence of cancer in the texture of that slice. And so we're not really worried about the prediction from these models. Instead, we treat them as a way to generate an embedding. So the embedding is the layer right before the output prediction. So this is the 64-element embedding vector. And so really what we do, we literally concatenate them. We just make a longer vector with the first 64 elements from the top network, the bottom 64 elements from the texture network. And then we can combine that into a multi-layer perceptron in order to make the final prediction. So there are multiple reasons why we do this. One of them, sort of, it adds a little bit more interpretability than if we were to leave out the textures at all, because the texture is a feature that we have more control over. It's something that we designed by hand. Additionally, it makes the network more accurate. So just intuitively, if you were trying to solve a problem and I give you two different types of information that are both relevant to the solution, then you'll probably have an easier time arriving at that solution. So in this case, we want the network to determine is there cancer in here or is there not, and we give them we give the network two types of information. And the result here is that we're able to perform segmentation in real time. And here is the graph of my result, where the white is the color-coded for cancer, the black is non-cancer, and here is the attenuation method, which I presented on the first slide. And you can see my method corresponds to the current existing method pretty well. And then what's interesting here, I could have left this as is, but I decided that I wanted to try and add even more explainability. And the way we do that is called gradient class activation mapping. If you notice, so I'll go back to the previous slide, I have a red dot right here on these layers. And so what that is that the convolutional layers extract representations of the layer before it. So you have 32 filters, then you generate 32 feature maps. And so here you have 128 feature maps that are of dimension 12 by 12. So what GradCam does is it takes the derivative of the output with respect to each value in the feature map. And so that, in a way, tells you how important each feature map was to the final prediction. And so then they do some global average pooling, get the coefficients, weight the feature maps by these coefficients. But the end result is that you can overlay these feature maps on the original image, and the red pixels, I can't switch, seems like I can't switch the slide. Thanks. So the red pixels indicate where the network deems to be important in the input image. So let's just consider the correct cases. So positive indicates presence of cancer, negative is absence of cancer. So in the texture, it identifies sort of the shape of the texture as an important distinguishing feature, which, if you recall from the slide where I showed you the training data, is indeed a correct way to look at the problem. But what's even more interesting is in the slice data, you have, in one case, the red pixels, or the red heat map covering sort of the top portion of the slice, and then in the other case, covering the bottom portion. And so really, what the slice data is, as you're going along the height dimension, you're going down into the tissue. And as I mentioned, for cancerous tissue, the optical attenuation is decreased. So what that means is that light is able to go further down into the tissue. So it really looks at, towards the bottom of the slice, what is the intensity of the pixels there, and that indicates to an extent what the attenuation of that particular region is. So in summary here, what we've done is that we've built a deep learning tool that is able to, in real time, which means as quickly as the optical probe is able to collect data, the neural network can process it, generate an attenuation map, a segmentation map showing the surgeon where the regions of cancer is while they are performing the surgery. And so, should have mentioned this earlier, but this is really important because if you're diagnosed with high-grade brain cancer with current medical technology, then that's really not good news. There's not too much that can be done to permanently cure the condition. But what we do have control over is whether your survival time is closer to several weeks or several months to several years. And in addition, during that period, what will your quality of life be like? And many studies link these two factors. The most important factor is the extent of resection, which means how accurately can you remove the cancer tissues so that you can delay recurrence and also avoid removing healthy tissue so that you don't accidentally inflict some neurological deficit. Okay, so the next application, I think it's probably one that none of you have heard of before because it's not something you would expect, but you can use X-ray beams to measure temperature in a substance. And the application here is called thermal ablation. You can non-invasively deliver heat into a tumor region. And similar to before, you want to deliver heat to the tumor while avoiding damage to the healthy tissues surrounding the tumor. And it would be great if we can construct a three-dimensional temperature map of the patient that tells you the temperature at each point. So currently what they use to monitor this treatment, they have to stick thermistors into the patient. So number one, that's invasive. The thermistors are also fragile, but I think more importantly, they only report temperature from a limited number of points, whereas here we're talking about a volumetric temperature reconstruction. The way this works is X-ray being just another wavelength of light, it also follows an attenuation law. So the attenuation is proportional to density. When you heat something, it thermally expands, the density changes, therefore the attenuation changes as well. So the straightforward idea is that we heat something by a unit degree of temperature, we observe its attenuation change. And here's some experimental setup that we used in the lab. So here's the actual detector setup. Here is the sample that we have. So in here is some sample material that we want to measure the temperature of. And here are some details of how we actually calculate the attenuation there. Not so relevant to AI, but just briefly, we have the projection when the phantom device is empty, and then we have the projection when it is filled with something. We take the difference, and the area is able to, from that area, we're able to derive the attenuation coefficient. But this is just some extra detail about the experimental setup. But what's the limitation of the approach and what's realistic in a clinical setting is that you're not able to calibrate your machine to the tissues of each patient. So you might know you're operating on a kidney, for example, but who's to say that the thermal properties of one patient's kidney is going to be identical to the thermal properties of another patient's kidney? So what we would really like to be able to do is to measure the temperature of a substance without knowing its precise thermal properties and even without knowing what that substance is. And so this is sort of the impact of what I will try to show here. So one thing we use that's different from traditional CT scanners is we use a technology called photon counting CT. So in hospitals, really the output intensity of the X-ray source, it's not one energy level, it's several energies. But in a clinical current integrating scanner, that doesn't matter. They just treat all photons the same. You add them all up, hence the name current integrating. But with a photon counting detector, really you have one extra dimension, that being time. So not only are you able to measure the energy of each photon, you can also resolve these collision events with respect to time. And so what that allows us to do is to say, when this photon hits, we know its energy, and then we can attribute it to an energy bin. And so that is able to treat the source more realistically to how it is, and it's a bunch of collections of different energies. So why is that important? Well, first, we claim that the attenuation mu of an unknown material can be expressed as a linear combination of the attenuations of in-base materials, the attenuations of which we know. So and under the condition that the sum of the volume fractions is equal to 1. So this is the idea of material decomposition. We want to be able to know the composition of the material without being told beforehand. And of course, you have n unknowns, V1 to Vn. You need n equations. And the only way you can do that is if you're able to measure the attenuation at each energy level. So if you have n energy levels, then you have all the necessary conditions you need to solve for your unknown volume fractions. And so the takeaway here is that photon counting CT, the spectral attenuation data you measure from it, encodes information about the tissue's composition. So here's some data just to show you some experimental data. As you increase in temperature, on the y-axis is attenuation. Increasing temperature causes a decrease in attenuation in all the different energy channels, so color-coded blue, orange, green, red. So the relationship is opposite in the orange channel due to an effect called Compton scattering, but that's not too important to realize here. So this is just some experimental data that this paper is also currently in peer review. And so here are some results. What I've hoped to show is, so how does deep learning come into this? How does machine learning come into play here? Well, so hopefully I've convinced you that so the delta mu1 to delta mu4, when you look at a change in attenuation, that encodes information about change in temperature, because temperature causes thermal expansion, causes decrease in density, causes change in attenuation. So here we have the information from the change in temperature. But up top, we have just the regular attenuation values at some baseline temperature, which hopefully I convinced you that in photon counting CT, when you measure different attenuations at different energy levels, that encodes information about the material composition. So now the neural network, knowing the material composition, as well with information relevant to material composition and information relevant to the temperature, is able to predict the temperature of an unknown material. And so that is precisely what we were able to do. We take 300 millimolar calcium chloride. So we trained the data on water, 50 millimolar, 600 millimolar calcium chloride. Then we tested on 300 millimolar calcium chloride, sort of as an interpolation. You can have 50% water, 50% 600 millimolar. That will give you your 300 millimolar. And we were able to predict with fairly good accuracy. But then we also try an extrapolation where a protein shake cannot be directly modeled in any of the base materials. And we still achieve high accuracy. Okay? And just briefly, I want to talk about some of the future work I'm doing. Can you play the video right here? Can you click the play button? Do you see it here? Great. So what I'm doing, so my next project at my lab at Hopkins is to analyze mouse behavior from their neural data. So we want to see to what extent can we predict its behavior? Can we predict when it's making a decision to turn? Can we predict how fast it's going? And so what I've done so far is, so we have a camera watching this poor mouse with an optical probe in its brain, running around this sort of box. And then we have a camera looking down on it, and we want to extract its location and direction at every point. So this is the diagram here, where the direction of the arrow is where the mouse is pointing, and the segments of this graph is color-coded to its speed. So this is my ongoing project, and I think it's more theoretical than the other stuff I worked on. And I'd just like to end with an acknowledgement to certainly my lab over here at Johns Hopkins, to Rensselaer Polytechnic Institute in New York, where I actually scanned the samples with their photon counting X-ray system, and of course, Rework for inviting me and providing the venue to deliver this talk. So thank you.